everyone, this is Heather Benson, and I am recording this video to explain how I did the written portion for exam 2 for Bio 375, and I'm going to do a screen share so that you can see my work. So let me just get to what I need to get to. Sorry, I'm on a different laptop here because I recorded this a minute ago and it did not work. I couldn't hear the volume on mine. Okay, well, here we go. So for problem number 1, it says, suppose an autosomal dominant polycystic kidney disease affects 1 in 750 individuals in the human population. For parts A through C of this question, please write your answer to 4 decimal places. So the first thing we do is we use the Hardy-Weinberg equation. What is the frequency of the non-disease allele in the population? Well, you can see I wrote this up here. The disease is 1 out of 750 individuals and is dominant, which will eventually correspond to the p-value. And the non-disease individuals are 749 out of 750, which is recessive. So for part A, what we do is we do q squared equals 749 divided by 750, and we square root both sides so that we can get the q value. The q value is this number here multiplied by 100 equals 99.9333%. And that is the frequency of the non-disease allele in the population. For part B, what we do is they want to know what is the frequency of the disease allele in the population. So we do 1 minus Q equals P, which is 0.00667 times 100 equals 0.0667% of the population has the disease allele. For part C, what is the frequency of the heterozygous individuals in the population? And so what we do for that is we use 2pq from the Hardy-Weinberg equation, which is 2 times p times q equals 0.001333 times 100 equals 0.133% of the population are heterozygous individuals. For part D, out of a population of 1 million people, how many heterozygotes are expected? And so we use the number that we calculated up here from the 2pq, which corresponds to heterozygous, and we multiply that by 1 million people, and that is about 1,333 people. Here's the exact value. Great. So for part number 2, it asks quite a few questions, and we're considering a DNA molecule. So I have some of my answers up here, but they're also down here, so we'll scroll down here so I can show you what I'm talking about. So it says the letter W above indicates the phosphate group. That's here on the end. The letter X indicates the deoxyribose sugar, which is deoxyribose because this is DNA, rather than RNA. The Y letter indicates the nitrogenous base, which is thymine right here. And the Z indicates a nitrogenous base as well. I'm assuming that this is guanine due to the way that the patterns are, because up here we have a cytosine, which would correspond to guanine, and the same pattern is down here, so I would say that this nitrogenous base here is guanine. Then it wants to know what the 5' end and the 3' end of each DNA strand are. So because of the location of the phosphate group here, we know that this is the 5' end, and this is the 3' end, but on the other side it's opposite because the phosphate group is here on the end in its position. So this is the 5' end, and this is the 3' end. Then it wants to know where the backbone is. And the backbone is the deoxyribose sugar plus the phosphate. So that is these portions on both sides here. Then it wants to know what portion gives DNA a net negative electrical charge, and that would be the phosphate groups. And those are here, these big black looking dots on both sides, those are the phosphate groups, and they give the DNA molecule a net negative charge. Okay, so moving on to problem number 3. 3 asks about linked genes in the fruit fly Drosophila. It says kidney bean eyes are K asterisk. E eyes are ebony body color. Then we have E star is normal, and CD, which is cardinal eye color, CD star, which is normal eyes. And in each of them, the one with the asterisk. So the first thing that we do is we set up the trihybrid cross. So it told us that there is a heterozygous female crossed to a homozygous male or multiple females and males. And it wants to know what the allele, the first part we need to do is decide what the allele order is for the heterozygous parent. So the first thing that we do is we look for the parental offspring in the table that they gave us with values. And that, what we're looking for is the 2 most numerous genotypes, or excuse me, phenotypes. And so for that, it's kidney, cardinal, wild type. Ebony, wild type, wild type. For that, we have the corresponding genotypes and corresponding frequencies. And so that gives us the answer to part 1. This is the allele arrangement in the heterozygous parent. The third thing that we do is look for the double crossovers on our values, and those are the least numerous 2 values. So what we have here is kidney, ebony, and wild type. Type and wild type. We have the corresponding genotypes and the corresponding frequencies. So the next part here, what we do is you take the allele arrangement in the heterozygous parent and set it up and switch the values until you get 3 different orders, and to determine which one is the correct order. So what you do is you line these up, and then you kind of do this double crossover thing here, and you look and see which of these lines up with the genotypes that we have for the double crossovers here. These do not, but these do. We have CD, K star, and E star, which is right here, and CD star, K, and E, which is right here. So we know that this is the correct order. So that is the answer to part 2. This is the gene order of the 3 linked genes. And so we rewrite that, and then we go on to calculate the map distance. So the first thing that we do is look at the map distance from one end to the middle. And so what we do is we line up what we calculated in the previous step, and we kind of do a cross this way and a cross that way. We get what's called single crossovers. And single crossovers for this are CD, K star, E, and CD star, K, E star. And that is 89 and 97 for those genotypes, or rather the phenotype shown as well. So what we do is we take the double crossovers from above, 8 plus 6 plus 89 plus 97, divided by 4,000 total individuals. And I got that by adding up all of the numbers that they gave us in the chart. You take that and you multiply it by 100, and you get 5 map units. Then for part 6, what we do is you look for the map distance from the middle to the other end. And so you can see we do it from the other end here. Get the genotypes. And we have the other single crossovers, CD, KE, CD star, KD star, E star, their corresponding frequencies. And we do the same thing as in step 5. 8 plus 6 plus 138 plus 128 divided by 4,000 total flies times 100 equals 7 map units. So then what we can do is we can put a linkage map together in part 7. And this is what we have here. We do the alleles on the end, 5 map units, the alleles in the middle, 7 map units, the alleles on the opposite end. And this is the answer to part 3. This is our linkage map. So the last step here is to calculate the interference. And so what we do for that is you do the 5 map units divided by 100 times the 7 map units divided by 100 equals 0.0035 times 100 equals 0.35%. And that means that 0.35% of the time a double crossover can happen. So then what we do is we need to determine how many expected double crossovers we have. So we have 4,000 total flies times the value calculated above equals 14 expected double crossovers. Then what we do is we take 6 plus 8 equals 14 expected double crossovers. This is what we calculated on the previous page. So the interference value, interference equals 1 minus observed over expected double crossovers. So we have interference equals 1 minus 14 over 14. Now 14 over 14 is just 1, so 1 minus 1 is 0. So the interference value for this is 0%, which is the answer to part 4. And this means that all of the double crossovers that we expected to happen did happen. And so that is it for this problem. So going on to our last problem here, number 4. It says that it has this syndrome that is a rare genetic disease inherited in an autosomal recessive fashion. So I've noted that here. The non-disease allele is capital D, dominant, while the recessive allele disease, or while the disease allele is recessive, lowercase d. Then we have the corresponding values that were listed. So there's 1 in 32,000 disease, people have the disease, and that will eventually correspond to Q. So we use the Hardy-Weinberg equation that I have listed up at the top there. And we do Q squared equals 1 out of 32,000. Take the square root of both sides to get the Q value. That is 0.005590. Times 100 is 0.5590%. That's the frequency of the D allele in the population. For part B, what we do, they want to know what the dominant allele frequency is in the population. So we take 1 minus the Q value that we calculated equals the P value, which is 0.994410 times 100 is 99.4410%. For part C, what is the frequency of the homozygous dominant genotype in the population? So homozygous dominant in the Hardy-Weinberg equation is P squared. So we take our calculated P value from above, square it, and we get 0.988851 times 100 equals 98.8851%. And that's the percent of the population that has the homozygous dominant genotype. So for part D, what is the frequency of the heterozygous genotype in the population? So the heterozygous portion of the Hardy-Weinberg equation is 2pq. So we do 2 times the p value times the q value equals 0.01118%, or 8, excuse me, times 100 equals 1.1118% of the population has the heterozygous genotype. And that's it for all the problems. So thanks for watching, and I will turn off the recording here. Thanks.